Again, this is the Functional Fungi webinar, and we're going to be discussing bioremediation strategies for brownfields. My name is Sebastian Harrison. I'm with the Center for Creative Land Recycling, and really excited to be joined by Danielle Stevenson, who recently got her PhD in doing case studies with this microremediation work. A little bit about our agenda before we get started. Just begin with an introduction, share some information about CCLR for folks that might not be familiar with our organization. And before we go really deep into microremediation, I just wanted to offer the larger context of bioremediation and what nature-based solutions can look like on brownfields. Then I'm going to hand it over to Danielle, who's going to talk about her work, some of the case studies, next steps for evaluating microremediation. We're going to talk about pilot studies and how you could further evaluate this work on your project, about CalRC, a conference that we're really excited about later this year. And then finally, we're going to end with about 10 minutes of question and answer. So if you have any questions as we're moving through the material, please feel free to put it in the chat or raise your hand at the end, and we'll try to go through your questions in order. Additionally, my colleague Sarah is going to be putting some helpful resources in the chat as well. So yeah, please, if you have very project-specific questions, it might make sense to set up a TA meeting after or reach out to Danielle, but we'll get into all that information in a little bit. So following the agenda, just talking about the learning objectives. Again, the context of microremediation, talking about in situ versus ex situ considerations, and that means, should it stay or should it go? Are we able to deal with the contamination in place on the site? Or are we going to have to, excavate the material and possibly haul it off to a landfill or some other type of ex situ solution? As we're talking about microremediation, this is a type of bioremediation, as well as this idea of nature-based solutions and how we can harness the power of nature, to heal nature and deal with contamination. Really the focus of this meeting is fantastic fungi and functional fungi. So we're going to talk about breaking ground research that Danielle's been doing. And then finally, we want to hand you some tools for piloting this technology on your project. And what are some of the next steps for that? I think many of you are familiar with CCLR, but just to really stress our mission that we are a national nonprofit dedicated to the sustainable, equitable, and responsible reuse of underutilized environmentally impacted properties. They could be contaminated. You could not know the status of the property. Really, the big message for you here is that CCLR is here to help. We work with all different types of stakeholders, from local government to community groups, to consultant organizations. And really, I think we do a great job of connecting the puzzle with all the different things that are necessary to revitalize a contaminated property. One of our biggest roles is we are EPA's Brownfield Technical Assistance Provider. And one of the key projects I've been working on has been our role with DTSC as a BTAP for the Equitable Community Revitalization Grant. And also the last part is we've been participating in more and more of the Region 9 EJ work. Today's speaker, so my background is I'm an environmental engineer. Been with CCLR for about 3 years. And before joining CCLR, I used to work in the chemical industry, bringing remediation projects to the field. So evaluating these different types of technologies and ultimately, putting together the bidding event and making the cleanup happen. I said before, but I'm really excited to be joined by Danielle Stevenson, who recently got her PhD. She'll introduce herself longer later when I hand over the mic in 5 minutes. But it's really been a joy to partner with her and to help share her work, because we need dedicated experts like herself to make innovative work like this possible. I first met Danielle through a panel we arranged through CalRC. The title being an ode to the Clash song, Should It Stay or Should It Go? So we put together a different panel to really just explore this idea of in situ versus ex situ management of contamination on brownfields. Again, is there a way to deal with the contamination in place? And this doesn't, have to be bioremediation. There's also chemical treatment, different chemicals we can introduce to the site. Maybe there's even a way to heat up the soil and create thermal treatment of the project. And this might be, opposed to the more traditional approach to site cleanups of excavation, offsite disposal, dig and haul, or ex situ management. So I think most people would agree that it would make sense to deal with the contamination in place if you could. I think, when you're looking at a construction site and you see these big, huge piles of dirt and you just, take measure of the number of trucks that might have to leave a contaminated property. If there was a way to deal with the contamination in place, most people agree with that. It would make more sense. It would likely be more cost effective and, would make sense from a sustainability perspective because there's less of a landfill demand and CO2 emissions. But despite that, dig and haul is much more common, and that's because with dig and haul we have some more certainty. We know that the material is leaving and the contamination might be leaving the property, and we have some more time certainty. But really, we put together this webinar and we explored this of how could we help resolve this clash? What are the things that could give people more certainty with innovative approaches like micro-remediation? So again, what are nature-based solutions? Again, it's this idea of what if we could use nature, to help nature versus introducing, brand materials to the system or the site. Are there things already there? Is there a way that we could work with the local ecology, microbes or fungi, to manage the contamination and ultimately make the property safe again, for whatever that use might be? And when we're talking about brownfields, we're often, talking about underutilized properties, and we want to find a way to make the property safe for people, whether that's a community garden, a park, affordable housing, really any type of end use. So examples of bioremediation. Of course, we're going deep into microremediation on this meeting, but it's important to know some of these larger terms that microremediation works with, such as microbial remediation, using bacteria, phytoremediation using plants, and then finally microremediation using fungus. And Danielle is going to go much deeper into this material, but just an important thing to consider is that these systems don't operate independently. With living systems, all three of these, are occurring likely at the same time. And not just that, those other types of remediation that we talked about, like the chemical processes, the geological processes, those are occurring too. So this means it can get very complex, but that just means we need to, update the tools in which we evaluate these sites. And I bring up all those processes that are working together to again help resolve this clash. How can we find harmony in this situation? And perhaps it doesn't have to be ex situ versus in situ or dig and haul versus things like microremediation, but we can find ways to pair them together for whatever might meets the site demand. And as we said, this will often make sense just for material management. That if you are hauling off 10% of the material to a landfill that you otherwise, would have had to, you might be saving millions of dollars on a big project. But to find out if these things are possible, it means that you have to, know what you don't know and pair with the experts early to try out this technology. And we're going to talk much more, but things that happen in the lab need to be tested out within the field to really evaluate if this is going to be the solution that works on your property. And, this is really just to stress my other point that this might lead to a much more complex conceptual site model as we're trying to understand the property. But ultimately, doing this extra work can save money on the property, make sense from a sustainability perspective, and minimize the community impact, not putting tons of trucks on the road, and instead supporting this nature-based solution. So thank you all for listening to me give this little bit of context about bioremediation and some of the things that we consider on our brownfields and trying to understand if this approach might work. I'm happy to now pass over the mic to Danielle Stevenson, who's going to go much more in depth on the topic. Thank you, Danielle.
Thank you. So I'm going to get my screen up. There we go. Thanks so much for that great introduction. I'm going to go a bit deeper into the potential of microremediation for brownfields cleanup specifically, and hopefully give a bit more understanding about fungi themselves. Why are we want to include fungi in brownfields cleanup projects, and some different approaches to doing so with some case studies from my own work. So I've been working around bioremediation and incorporating fungi into bioremediation for about 12 years or so, through a small mycology business where I was growing fungal inoculum and working with universities and governmental agencies and community groups to implement research and development of mycoremediation projects, which led me into my PhD in environmental toxicology, where I really wanted to explore scaling and field testing, further field testing of some of these methods. So it's so exciting to me that this webinar is even happening, and there's potential to start to work together to apply and field test more of these methods. So just to first of all define mycoremediation and place it within the category of bio or biological remediation. So biological remediation involves working with living organisms, plants, bacteria, and fungi to remediate or address contaminants in the environment. When we're working with plants, that's called phyto-remediation. When we're working with bacteria, that's called microbial remediation or bioremediation sometimes. And of course, we're focusing today on the fungi, mycoremediation, which has long been sort of overlooked. And so mycoremediation involves working with fungi to degrade organic contaminants or enhance phytoextraction or removal of metals. We're going to define all of those terms in there next. And there's a lot of potential within this space. So we can work with fungi also to address hazardous wastes. But today's presentation is really focused on remediating contaminated sites in situ or in place with fungi. And just some basic fungal ecology and basic info about fungi, so we're all on the same page. Quick question too. Can anyone guess? You can put it in the chat. What living thing is showing in my background? You can guess. I will clap for you. So, yep. Okay, I see some guesses coming in. Oh, yeah, somebody got it. Mycelium. Okay, so that's a perfect segue. So I wanted to define mycelium, because when we're talking about mycoremediation, we're working with fungi, but the word mushroom gets thrown around. It might be a little confusing. I just wanted to clarify that mushrooms actually refer just to the reproductive part or the fruiting body of certain fungi. So not all fungi even produce mushrooms to spread spores and reproduce, but some do. And typically, when we say mushroom, we're referring to that reproductive part that's shown here. The mycelium, however, is the actual living fungus. It's the sort of vegetative body. It exists as a web of thread-like structures that's interacting with its environment, taking in food and water, and so on. And so there's a diagram showing what mycelium looks like here. And then this is an actual picture of mycelium. It's typically growing underground or in places we can't see it. So it's helpful to define. So when we're talking about remediation with fungi, we're working with the living organism, the mycelium. And so just to create a frame for how I'm going to move through this presentation, I want to set up some basic principles of mycoremediation that I've defined based on my work in this field over the past decade or so. And so you'll look for the icons as we move through to help keep track of where we are in our discussion of these and how these case studies and so on relate to these principles. So the first principle I'll go through is design your mycoremediation project or your bioremediation project based on the contaminants present and the site conditions. Next, I'd suggest picking fungi that are already adapted to the contaminants, right? And the site conditions. further, we'll want to, as Sebastian brought up, we want to mimic nature. It just makes most sense. I'll go through and explain that a bit more. But in nature, we see diversity leads to resilience, and there's an ecological principle called succession that I'll explain a bit later as well. And then finally, really importantly, you want to give the fungus what it needs. And so to understand what a fungus needs, you need to understand its basic ecology. How does it live? How does it get its food? And where does it live? And so we'll be going through some of that as well. Okay, so in terms of the first principle, choosing your strategy based on the contaminants present, I just want to differentiate two main categories of contaminants. And this is really important because this will drive the design of your strategy, right? So the first category I want to talk about are the metals. They're also known as elemental or inorganic contaminants. You've probably heard of lead and arsenic. These are examples of metal contaminants. The thing about metals is they generally can't be broken down. So your remediation strategy will involve moving them around. So you can either try to bind or stabilize them in soil, extract them, or react them to be less mobile or toxic. Our next category of contaminants are organic contaminants. And so these are carbon-based. It means that they were manufactured or derived from something that was once alive, containing carbon. And so examples of organic contaminants would be any type of petroleum product, like diesel, persistent organic pollutants, PCBs or dioxins as examples, agrochemicals, and so many other contaminants. So now in terms of picking a fungus that can help address these contaminants, there's general categories of fungi based on how they live and thrive in nature that will tell us a lot about how we might be able to work with them for remediation. So the first major category of fungi to know about for remediation are the decomposers. So these are fungi that in nature break down dead stuff, mostly plants. Mostly they're trying to get that cellulose or lignin. And this is how they get their food. So they actually metabolize, a dead tree using these enzymes that they excrete into the tree. And so the thing is that these decomposer fungi have the potential to degrade petrochemicals and other organic or carbon-based contaminants, right? Using those same enzymes. And the reason they can do that potentially is because, a lot of petrochemicals and a lot of organic contaminants are just made of dead plants or dead stuff that's been compressed for a really long time. Our next category to know about for remediation are the endophytic fungi. So these are fungi that live inside of plants without causing disease. They can act as decomposers of organic contaminants, or they can also really help plants grow and enhance phytoremediation success. Another category that's good to know about for remediation are the mycorrhizal fungi. So myco meaning fungal, rhizal related to roots. These fungi live in mutually beneficial relationships with plants through their roots, where they trade access to nutrients and water and stuff to get their food. And so these fungi can be utilized to enhance phytoremediation success. And why fungi? Well, they show up all over the world, wherever there are toxic sites or toxic materials. So I have just a couple examples here. There's been plastics, floating in the ocean that have been found to have a number of common decomposer fungi and common fungi that live in soil and grow as molds. Even Chernobyl, the melted down nuclear reactors, was found to have a bunch of these common like mold fungi growing in there, actually working with the radiation. Yeah, electronic waste has been found to have fungi growing on it. That can be used to strip metals from electronic waste and recycle them. And even pure oil tailings have been found to have endophytic fungi growing in there, decomposing the oil tailings and also helping plants grow. So just a little more detail on some of these fungi that are especially applicable in remediation, getting into some really specific types. Want to tell you a little more. We mentioned there's mycorrhizal fungi that connect to the roots of plants. Well, there's a specific type of mycorrhizal fungus called arbuscular mycorrhizal fungi. I know it's a mouthful. I'll refer to them as AMF moving forward. So these fungi have been shown to potentially help plants extract more metals from the soil and thereby enhance phytoremediation success. Got a diagram shown here of just, the plants and their roots without the fungus. And then you can see, even in this depiction, that these AMF networks greatly expand the root zone of the plants. So expanding their potential to reach and pull up different metals. Other fungi or decomposers, such as the oyster mushroom or Pleurotus austriatus, have been found to be able to degrade organic contaminants such as diesel and PAHs and more. And so what happens in nature, though? We don't see in nature a single species or even a single kingdom of life, right? We see a diversity of different types of fungi and bacteria and plants, and we see that they each have a stage in an ecosystem at which they play a bigger role. And so I've broken apart those stages here just to show how when you put these approaches all together, you can address the complex mixtures of contaminants that are often found on brownfields, right? Where similarly, you don't have just a single type of contaminant, you probably have a mixture. You probably have lead and diesel and, something else. So in nature, we typically have these beneficial decomposer fungi breaking down carbon, but they're able to break down stuff that's too complex or too hard to break down by bacteria and by secondary decomposers. So they start that process off, and as they do so, as they're breaking down, whether it's a dead tree or a complex organic contaminant, they're creating soil as they do so, and now facilitating a process where, as soil is created, plants can grow. When the plants have these arbuscular mycorrhizal fungal partners connecting to their roots, it can help them pull up more metals. Metals are like nutrient elements, and so plants take them up anyways. Some plants are extra good at that, and these AMF may really help them do so. And then once those beneficial decomposer fungi have started creating that healthy soil and breaking stuff apart, now bacteria and secondary decomposer fungi can come in and complete the process. And so in my years of working on this, I found the most successful remediation approaches mimic nature. And so another way to say that is just the joint implementation of myco with other remediation strategies tends to be most advantageous. But we've been missing the fungal part, right? We've been missing the fungal part. So trying to add that in now. Okay, so how to incorporate fungi into remediation projects. I'll go through some different approaches and best practices. And there's three main approaches that I'll go over today. The first one is that you can find fungi that are already growing on a given pollutant or waste or contaminated site, and then amplify them in place. So give them what they need, and help them do what they're already doing faster, but more efficiently. Another option is that we can survey contaminated sites, or landfills, or places where these pollutants are found, and figure out what fungi are growing there, and then screen them for remediation capacity for use on other sites or wastes. There's an example here from a paper by Thomas et al. In 2009, where they collected fungi from the local environment, isolated them, grew them out in bulk, and then used these local fungi to then implement their water filtration. I believe it was a storm water filtration study, which was highly effective. I also have employed this approach before. I did a study of 7 contaminated sites, brownfields and Superfund sites around LA County and Southern California that are shown on this map. And I identified all the fungi, microbes, and plants that were sort of volunteer cleanup crew on these sites, right? Nobody planted them. They weren't getting irrigated or fertilized or anything, and they were growing on their own. So typically, if, like, going back to Chernobyl and these other examples, if a fungus is growing somewhere, it's probably because it can eat something there, right? So this is a great option for identifying fungi and other living organisms that are not only locally adapted to the environmental conditions, but also able to play a remediative role. And so this is a lot of Latin names. Don't worry about that. I just wanted to share some exciting news that through this study of these 7 contaminated sites in SoCal, we identified previously untested fungi. So these are taxa of interest for bioremediation, as well as already known and tested fungal remediator species. And so super exciting. Just want to demonstrate that this approach is a really effective thing to do regionally to identify fungi that can grow without a lot of extra resources, and could be good candidates for remediation of what's found on these sites. So a second approach we can take for mycoremediation is acclimatizing a fungus. So you say you've done the first approach, you identified some fungi that you want to test for in remediation. You can actually then acclimatize them to be even more efficient and rapid at either metabolizing, so breaking down, or tolerating a given pollutant. And this is possible for both metals and organic contaminants. So the cool pink and green image is showing a really common fungus. It's like a mold species that was exposed to increasing concentrations of arsenic, such that it could actually grow faster and better. It got used to, essentially, these higher and higher concentrations of arsenic, such that arsenic came to actually enhance its growth at high concentrations. And then this is a picture for one of my own studies in the past, where I was acclimatizing a Pleurotus species to metabolize a particular lubricant, so petroleum product with Teflon added in. And you can see the fungal mycelium there is growing rapidly towards the pollutant, right? So it's hungry for it. And that's sort of a method that can be used to optimize remediation. And the third approach that we can take for mycoremediation is just introducing a fungal species or more known to play a needed role. So how do you figure this out? Well, you can review the literature, see what other studies have been done, and identify fungal species that you have been shown to be able to degrade or deal with the contaminants you have on your brownfield. So I'm going to go through now my some of the findings from my dissertation work at UC Riverside. We did a phyto-mycoremediation study on 3 brownfields in LA County. And we actually compared, as I'll get into, we compared these approaches. So we compared just amplifying the fungi that are already in place with introducing known fungal remediator species. So like I said, we tested this on 3 brownfields that are shown here in partnership with the Bureau of Engineering, Sanitation Department, Brownfields Program, and a community land trust. And these sites all had mixtures of metals and organic contaminants. You can see they had some similar contaminants and some different ones. And they all were intended to become some type of park or green space and or community space, housing, community center, and so on. So the bioremediation treatments we tested over a 12-month period included the top native plants that we identified in that first survey I mentioned that were growing on contaminated sites in the region and taking up the highest concentrations of metals of concern. We compared that with a known remediator species plant that was a non-native plant. And then we tested these plants alone and in combination with these 5 different fungal remediation treatments. So one was just the indigenous mycorrhizal fungi only, so no inoculum. We compared that with a commercial AMF inoculum. And we had a native decomposer fungus in the form of spent blocks from a local mushroom farm, compared with indigenous decomposer fungi only that we stimulated by adding wood chips and water. And then we tested these fungal treatments together. So both types of inoculum, the AMF inoculum and the P. austriatus inoculum. And we tested this with and without irrigation and ran it as a rigorous scientific study with lots of replicates and controls and so on. And I mean, overall, I'll get into the details, but we did find that mycophytoremediation with native plants, AMF and P. austriatus, showed a lot of promise for Southern California brownfields remediation. So specifically, we found that plants and fungi reduced soil metal concentrations significantly in just a 12-month period. So we averaged results across the three brownfields in the study and found that compared to control or untreated plots, there was significant reductions in zinc, copper, lead, arsenic, cadmium, and chromium. So that's really exciting news. I was delighted to learn that the arbuscular mycorrhizal fungal inoculum, something that's easy for folks to access and purchase, significantly increased plant survival and biomass and was the most significant contributor to plant metal removal from soil. So enhanced phytoremediation of metals. Interestingly, we also found that the oyster mushroom inoculation significantly increased lead removal by plants. So that's good news because lead is a really difficult metal to remove from soils effectively and is also everywhere and highly toxic. And then in terms of the organic contaminants, we are showing in some of these pictures and images. These are the spent blocks from the mushroom farm. This is a picture, again, of the, like my background, of what was happening underground, where the white mycelium is growing through the contaminated soil. And here's a cross-section of that treatment. And when we pulled together all of the different types of organic contaminants, so diesel, gasoline, solvents, PAHs. There was an overall 99.75% average reduction in when treated with P. austriatus. And we're showing here just a graph. So in green, you're seeing the concentration before, and in light green, you're seeing the concentration after. Here we're seeing the treated plots, so where we put the mycoremediation treatment in compared to the controls. And you can see that that in the treated plots and the mycoremediation treatment, pretty high concentrations of these petrochemicals were reduced to almost nothing in the 12-month period. And this is extra significant. I'm not sure how many folks have worked on brownfields that have been sitting around for a long time, but it's extra difficult to, it's thought to be extra difficult to bioremediate aged contaminants that have been there for a long time. So this was exciting news. And I'll just end on some of the mycoremediation barriers and gaps and opportunities that we might consider and explore together. So first of all, there's been very few fungi tested. So for the myco nerds out there who know the different types of fungi, you might notice, yeah, it was mostly white rot decomposer fungi that have been tested so far, but even like very few species have been tested within there. There's a lot of room, especially seeing the types of fungi that are showing up in contaminated places. It's actually not so much these white rot decomposer fungi. We're seeing a lot of mold and a lot of different types of fungi. So we need to test those, explore more diverse fungi in remediation, especially those adapted to different environmental conditions. So for those who are paying close attention, you might have noticed that in my results from looking at fungi growing in SoCal on contaminated sites, you might have noticed that a lot of them were so-called extremophiles, meaning they can survive in extremely harsh conditions, like very salty, very hot, very dry, things like that. So we need to test more fungi, basically. Another barrier to mycoremediation is that there's a lack of scalable inoculum production and application methods. So it's hard to grow enough inoculum. We need techniques for that. And then a lot of the techniques we have aren't really suited to scaling up to the scale of the problem, like a whole site. And so there's an opportunity here to develop not only scalable inoculum production, but that's why I explored using mushroom farm waste as a inoculum source, and then exploring other methods of introducing inoculum or amplifying it in place. There's also some regulatory barriers and a lack of funding for some of this. So there's a real opportunity here to remove regulatory barriers to field testing new technologies and increasing funding supports and timelines for these explorations. Just to emphasize what Sebastian already said, there's very few field studies of mycoremediation, and this results in a lack of understanding of how these methods work in the complex and heterogeneous soil environments where they're needed. And so because we've done a bunch of testing, in labs in very controlled and perfect conditions, there can be gaps in field performance when we apply what we saw in a lab into a messy and hot field, brownfield condition. So let's definitely encourage and undertake more field studies on contaminated sites, especially in understudied climatic regions. And finally, there's gaps of understanding about fungi in general and how they live, and how we only know like 5% of the fungi that are expected to be present on Earth. You can't really study mycoremediation anywhere or get trained in it. So it means there's also a lack of skilled mycoremediation practitioners and expertise to be, running these studies and applying these methods in the field. So there's a real opportunity here to develop institutes for studying and training and educating people in these methods. And yeah, with that, I'll thank you for your attention and pass it back to Sebastian to wrap up the presentation.
Awesome. Thank you so much, Danielle. That was really good. Okay, just give me a second to share my screen. So I'm going to go fairly fast so that we have enough time for question and answer. I saw a lot of great questions in the chat. I want to make sure that people have time to ask Danielle. But I think a lot of people, the big question coming up, because we think about our projects is will it work? Will this work on my site? And the big answer as an engineer or scientist will tell you is it depends, right? Danielle just brought up the difference between the lab and the field. To really know if mycoremediation is going to be a remedy for your project, you're going to have to conduct a pilot study. And that means, building the right type of team, recognizing that you might not have the experts currently on staff or do this work. You might have to reach out to a university or a consultant like Danielle to help, craft this type of project. And the first things that, she or anyone else is going to ask you is about your project. Some of the useful information that we need to know, things like the size and the types of contamination. So here are some of the steps that Danielle outlined, which I think are really basic. But first things first, to an outsider, what is the site? We have to understand what the site is, test the soil, water, waste on the project to understand the contamination that we're dealing with, address what the current research is for approaching that type of contamination, if it's been done before and with what type of species, build a team and set our goals. And, the goals, our treatment goals for the project are going to be dependent ultimately on how the site is going to be, be reused. So make sure you're having that discussion early. There were some great questions in the chat about this, but where does the contamination go? We have to be very smart about not eating things that are growing on contamination, like oyster mushrooms growing on top of metal contamination. And that means building a plan, a waste management plan to dispose of any toxic byproducts, understanding where plants might be uptaking this material as well. And this really just stresses the point of working with an expert. And if you are, really addressing contamination, not trying to undertake this work by yourself. And again, this speaks to just protecting yourself, to wear proper PPE, when working on brownfields or contaminated sites. Pilot studies can take time, just like field work does. So you're going to have to maintain and monitor the project and think about that early, about what your testing processes might be. And ultimately leading to this evaluation of, will mycoremediation work on the project? What type of time frame? Or how might we pair this with a more complex remedy? But really, as you see this slide, to truly implement mycoremediation is going to take some time, some process, and it's important to build the right team. So please reach out, with your project specific questions. We can't attempt to answer them all on this phone call, to really get into the weeds or into the soil. But we want to hear from you. We want to hear your projects, and we want to support this type of research and innovative remedies. So just the last thing I'm going to mention before we jump into question and answer. CCLR is really excited to bring the California Land Recycling Conference back to Carson this year. If you enjoyed this talk, if you'd like to hear similar talks as well as just network with other professionals in the revitalization and brownfield redevelopment space, please join us. We would really love to have you there. And as I said at the beginning of the meeting, property redevelopment, brownfield redevelopment is complicated. There's lots of different stakeholders working from local government, technical experts, regulators, and it's conferences like this where all the pieces of the puzzle can come together. And we've really seen great teams and great work happen because of CalRC. So if you're curious, please join us. But with no further ado, we're going to hop into question and answer. And here you can see Danielle's contact information. if you would like to reach out to her, as well as some of the CalRC information. So I'm just going to hop into the chat and answer some of the questions here. But if we have time and you have preference, you can raise your hand as well, and we can try to do a couple questions via mic as well. The first question was from Anne, which was just about PFAS and whether they were organic or inorganic substances, Danielle, when you were sharing sort of that basic overview. I shared that they were synthetic organic substances. Unfortunately, PFAS is really hard to break down. But there has been some interesting research from UCLA and other labs about how fungi might be able to deal with this contamination. Have you worked with PFAS at all?
Yes, I did. So the training approach was actually with a Teflon, which is one of the PFAS chemicals. It is an organic contaminant, so it's carbon based, but it's extra hard to break down because yeah.
Yep. So again, we might have to consider how long we have to remediate the site, or maybe like carbon treatment could make more sense, but that's not to say that there isn't potential for microremediation PFAS. Next question from Mark. I thought this was really interesting because I hadn't thought about it this way. Danielle, can lower animals be used for bioremediation? Invertebrates, earthworms, insects, ants, termites, fly larvae. I brought up that
Absolutely.
obviously earthworm. Oh, go ahead. Yeah,
Oh,
please.
sorry. I just jumped in. I love this question. Absolutely. I don't remember if it was in some of my diagrams about mimicking nature, but absolutely. And a lot of my studies do include worms or yeah, some type of worm, but also termites and wax worms and all kinds of different invertebrates are play an important role in remediation as well.
Exactly. The biological system is working together, right, with all those wonderful things that are happening in soil. And when we're undertaking microremediation, it's not like all those bugs or insects are going anywhere, and they're all part of the system. Um, Next question is a bit specific from Karen, so maybe we'll follow up unless you know offhand. Is there a published reference for the work with the 7 sites that you brought up?
Oh, that's my work. I'm trying to publish it, so look for it soon. We're trying to publish, of course, in scientific journals, all of the findings for the studies that I referenced.
Fantastic. Yep. So please just stay tuned if you want to be aware of the publishing of Danielle's research. Um, Next question from Carl. I think you addressed this briefly, but it really is a good one. It came up later again. Um, do the fungi themselves uptake contaminants and contain elevated levels of concentrations of contaminants, metals, etc., that would need to be handled, disposed of appropriately following remediation?
Yep. So the mushrooms. So if you're working with a fungus that forms mushrooms, there's some I've always tested in my studies, but yeah, they can tend to accumulate or even hyper accumulate metals, which again don't typically break down. So if there's lead in the soil and you have you put in a decomposer fungus that forms mushrooms, and it forms mushrooms, it's likely that there will be metals and potentially other contaminants in those fruit bodies. So my projects always include a plan for harvesting any byproducts of the remediation and containing it, and concentrating it so it can be disposed of as a hazardous waste. And that's true of plants that are used to remove metals from soil, as well as mushrooms. I will say, however, that again, typically with remediation, we're working with the living organism, so the fungal mycelium, and mushrooms may form, but they don't always form. And again, not all fungi, like a lot of the fungal molds and stuff like that do not fruit mushrooms. But that's a great question.
Yep. Yeah, and I think obviously the classic science answer is it depends, but it's a phenomena that you have to be very aware of, and even on a property, sequencing off or labeling where plants might be uptaking levels just so that no one, consumes or does other things with those materials, right?
Yeah, and I'll just add in case folks are thinking like, oh no, well, is that any better then? I will say, having worked on many, many, many large contaminated sites, it is better because you go from a huge area being contaminated to, a certain amount of biomass, fungal or plant biomass, that contains the metals, which aren't going to break down anyways. Once you remove those, you can either incinerate them or sludge them, and that biomass becomes reduced to be, so you go from, again, a huge contaminated site to a small amount of concentrated sludge or ash, which can be so much cheaper and easier to dispose of that than all of the soil on that entire site.
Well said. Yeah, you're able to accumulate it. And again, we're ultimately able to, limit the exposure pathway or understand how the contamination could impact other people. The next question is a bit specific, so we might have to follow up, but maybe you recall. Was the chromium reduction that you talked about a measure of total chromium or a particular species? Or maybe it was like a transformation?
I the number I showed was for total chromium, but I did do entire studies on, so one of the field sites, and I did a greenhouse study on hexavalent chromium, the most toxic and mobile form of chromium. And we did find that these fungi, both types of fungi we tested, do reduce the chromium. So thereby reducing the toxicity of the chromium that's on site.
Thank you, Danielle. Okay, next question. If oyster mushrooms are good at picking up lead, are the mushrooms disposed of separately? Is it right to assume that they cannot be consumed? So this was the similar question I talked about if you want to respond.
Yep, don't consume them. Get your mushrooms to eat at a farmer's market. And then, yeah, I don't ever consume mushrooms used in remediation. Collect them like we discussed and dispose of them properly.
Yep, no, and that's even why it's important with culinary mushrooms to make sure that they're grown and sourced responsibly, because sometimes people can grow mushrooms on, former waste and things like this. So, yeah, like I mentioned before, it's just something to be aware of, that contamination, can be bioaccumulated. It doesn't mean that these things can't be used. It just means that there could be a waste to manage, after the project. Um, Karen has a question, and maybe you can answer in a more general sense because I know this is obviously project specific. Um, to what depth are metal and petroleum concentrations reduced? So wondering about like the depth of this technology could be applied at.
Typically, it's most effective for shallow soils, so the top two 2 to 3 feet for sure in a short time period. However, you can use engineering approaches such as some trenching that we did utilize in one instance, and or again, major advantage of working with some of these fungi and some of these native plants that have the capacity to grow deeper in the soil. So typically, I can answer in one sentence, just the remediation can occur to the depth that the organism you're working with can grow, right? So it depends, but definitely effective in a short time period, and like such as a year. In the top 2 to 3 feet can go much deeper though as well, and can engineer to increase depths as needed.
Totally. And as the, ecosystem facilitates, right? I think Danielle showed like the tree root system, how deep that might go. Again, there might be micromediation or would be micromediation occurring all along that line. So it could be potentially very deep, but if we were, just applying or implementing the system ourselves, we could be more limited, to the upper layers of the soil. This question is similar, but you might have a deeper perspective on it. How deep can the mycelium reach in the Veda zone? Will it extend through the capillary fringe? And are certain soils like clays more challenging for mycelium to grow?
So mycelium can grow very deep. There's reports from like the deserts of Southern California of mycelium growing 30 feet or deeper into groundwater to pass it back up to their to their partners and networks. So they can grow very deep. They can certainly grow through the vadose zone. Yeah, I mean, yeah, there can be differences depending on the soil type. In terms of remediation, that's why I would really love to see these methods tested on just again, more field testing on different types of soils. The 3 sites I tested on in my dissertation had different soil types, and we did notice differences. So yeah, we did notice differences, not as much in the fungal growth, but definitely in the, you know, soil type definitely affects the contaminant, either extraction or degradation.
Yep. And something like soil type is going to impact like all of the biogeophysical processes at the project. So that's why it's important to understand your conceptual site model. But like the easy answer, are certain soils like clays more challenging for mycelium to grow? Yes, they're more challenging for anything to grow. But they also might be more challenging for the contamination to be transported as well. So it all ties into that larger model of the site, right? Um, a definition question for you, Danielle. What is inoculum?
Oh, that's the source of the fungal material. So, um, does that make sense? So it's
Got it.
mycelium. Inoculum is typically mycelium. Sometimes it's spores. It depends on the species, but it's the material you introduce to your sites to get the fungi in there that you want to be able to grow and do the remediation.
Oh, that's awesome. That makes perfect sense. Um, we are coming close to the end of the hour. We're going to keep going with question and answer, but if you have to leave, please go on and feel free to email your questions if there is something that you're interested in following up about. Again, we would love to hear from you if you attended, what topics you'd like us to do next. Perhaps understanding how microremediation could be used in wildfire recovery, a follow-up on this, or another topic in bioremediation. CCLR would really love to hear from you on these types of webinars. Um, okay, next question, Danielle. Thank you again for staying a little bit later. How long do these mycelium live on in the subsurface?
That's really variable. Sorry, I can't answer.
Probably the
It's really
most, right? Because
dynamic.
it's so general. Yeah.
Yeah.
So I think some mycelium might have like a really short type of life and things like this versus what would be like the longest time that you would say one mycelium could live for?
Well, they can keep growing as long as
Yep.
there's food and suitable conditions. So, they can, spread spores and find new areas to get more food and keep growing. So they're pretty resilient, but it's just dynamic. So yeah.
Well
Um,
said. Now, and you brought up
yeah,
a site like Chernobyl or something like that, where you can
yeah.
see that life finds a way, and that after everything's gone, the fungi might still be there. Um, but here's a fun specific question, not related to metals. Is there a way to reduce harmful bacteria like E. coli from a sewage spill or parasites in the soil with microremediation?
Absolutely. So I've actually worked on a bunch of projects like this, and there's some neat field studies out there, including the one I referenced earlier from Washington. Um, yes, so E. coli and parasites are also pathogens or harmful to a lot of these fungi. So they're actually really good at dealing with them. So there's a bunch of neat work on that, both for water, contaminated wastewater filtration with these fungi, and in soil environments. It's kind of cool. A lot of these fungi, right, they typically eat dead stuff, but they will also catch and eat nematodes and inactivate E. coli and things like that. It's kind of cool.
Yeah, you're right. And maybe pair with the tree to protect the tree or some other type of living organism, from the harm of the parasite or what have you. Um, yeah, I know Paul Stamets is doing some interesting work with like beehives and how they might work with mushroom species and things like that. Okay. A fun question from Christina. She's wondering, Danielle, could you imagine scaling to an elementary community school footprint?
That's actually how I got into this work initially. I have done this with public schools, and I'd be happy to to work with you on that. I actually love working in schools. We have actually, yeah, and we've worked at elementary schools, but also high schools, and we have a workforce development youth program specifically for bioremediation, where youth get a certificate in this stuff. So please do feel free to reach out if you wanted to collaborate on something like that.
Fantastic. Yeah, and I think that is the type of like project specific thing. It'd be best to reach out to CCLR or Danielle. And yeah, we can help grow out the idea. Sarah was looking for more information on this and PFAS. So that might be a good follow-up for us, unless there's anything else you want to add on the PFAS topic, Danielle.
No, I've worked on it. It's definitely, I've seen fungi degrade PFAS time and time again. The issue with PFAS is more figuring out whether mycoremediation is a suitable technology for the environmental media and scale. So PFAS is literally everywhere in the water, in the soil, in our blood, in wastewater. And so it's just figuring out, okay, we know fungi can degrade PFAS, but can we apply fungi in like all the soil everywhere, or you know what I mean? So there's some scaling and applicability constraints and considerations there, but it would be great to see more exploration there.
Yeah, exactly. And this might be where, micromediation has to work with like traditional, like a carbon treatment system or like hotspot dig and haul removal of the high levels, but there's an area on site where there's ongoing, um, you know, degradation of PFAS with micromediation. So I think that's a cool question. I think, just with how hot the PFAS topic is, there's going to be more interest and more focus on just that. And hopefully, people exploring like the nature-based solutions to PFAS management. Um, Sharon asks a question about developing real studies and field work that I think is best for us to follow up on like a one-on-one basis, but just want to again reiterate that message that CCLR is here to help you figure out what a pilot study might look like for one of your projects, and how this work could come to be. Several folks have asked about the recordings. Yes, there will be a recording, so don't worry. I know that there was a lot of like text heavy in studies, so we'll make sure that you can get the references as needed. Um, I'm just going to go down to now Daniel's question. Um, Danielle, have you seen the use of fungi remediation in conjunction with chemical in situ strategies, such as ERD and ISCO, etc.?
I think it's a great idea to explore that. I have talked with some companies doing chemical strategies to look at different combinations. I have done some testing of a small amount of testing myself, but I think this would be a great area to explore. Yeah, and just in general, looking at where there can be synergistic or additive, like basically how to optimize, like where is microremediation most appropriate? How can we optimize it to achieve, cleanup goals for these sites? So yeah, I think it's really promising and would love to see that happen more and help explore that.
Well said. Like no particular projects are jumping out at me for the fungi, but I've definitely seen phyto-remediation paired with chemical in situ strategies. And that has a little bit more industry acceptance. So microremediation is playing catch up a little bit. So I think we'll see more of these things, just to reiterate this message that the biological system is working at the same time as the chemical, the physical, the geo. So oftentimes it could be the more complex remedy that can fit your project. Um, oh, Carrie, I'm sorry for misnaming you before, but we'll make sure that we connect you with CCLR. Another question from Christina. Um, I think she's just asking for help, but she's wondering, have you done any work with air quality and micromediation, Danielle?
I haven't really. I have studied it, and I have heard of some um tests that were done, not just solely with fungi, but with, you know, um, like moss and lichen and different different species that are more known to filter air. So far as I know, I haven't heard too much about air filtration with fungi yet, but it'd be another neat area to explore for sure.
Yep, yep, well said. And this might be where trees might have a really big role as well, within air quality and air filtering, depending on the project. Um, Technical question for you, Danielle. Are you aware of any research regarding the treatment of dioxin or furan contaminated sediments and sludges?
Yes, yes, there have been some studies into that. That found, I even think that Pleurotus species and a number of other common, or at least most widely tested decomposer fungi have been shown to be able to degrade dioxins and furans in sludge and sediment. So you could check the literature for those. I'm not remembering offhand how many of those were pilot studies, like in situ. But I definitely know that there are fungi that are capable of degrading those, at least in bench scale studies.
Well said. And I'll just add a note because I introduced this idea of in situ versus ex situ to the meeting. The line between the two can be a little bit blurry. You know, we might be digging up the soil on the material, making a pile, sort of an in situ mix with ex situ because we are taking it out of the ground. But that might be the best way, you know, to have the microremediation perform or any other type of treatment. But that could still make a lot more sense than, you know, hauling the contamination potentially hundreds of miles to a landfill, right? Um, okay. Hope answers the question. What is proper disposal? Just landfill it, and that could be appropriate. Again, it depends on the type of waste, the types of levels. But do you have any more insights on disposal you want to add,
Just
Danielle?
that you would test it. So typically, I mean, with any remediation project, right? The same thing. You would test it, figure out what the levels are, what contaminants are still present, and then that would determine how you need to dispose of it. So in the past, I've had to pay for the sludge to be disposed of as a hazardous waste, a hazardous waste landfill, but sometimes it might be acceptable in other types of landfills. So you just have to test to find out.
Test and plan early. Plan early about the types
Yeah.
of facilities so you understand what their testing criteria are and you can design your project around that. Hazardous waste disposal can be very expensive and very tricky. That's part of the reason why something like microremediation could be such a slam dunk to begin with. Um, but yep, these things take evaluation. Um, Anne has a fun message in the chat for you, Danielle. I just want to reiterate it. Thank you. You are a Myco hero. Um,
Oh,
yeah, as an environmental engineer that loves this material, you know, we need people like you to take the deep dive and push this work forward. So yes, you are a Myco hero.
thank you, Anne. Anne's a pretty cool Myco hero too. And thanks everybody. Yeah, feel free to, I do encourage folks, feel free to reach out. This is my passion. So if you want to work on something together or yeah, access some of the resources that I'll be publishing, don't hesitate to be in touch.
Awesome. So scanning through, I think I got to all the questions. So again, just thank you everyone for joining us and everyone that helped put together this webinar. Hope everyone has a great day. We will send out the slides, and please reach out if you have any questions or big topics you'd like to hear from us on. Any closing words, Danielle?
No, thank you again. Thanks for all the great questions too.
Awesome. All right, everyone, have a great day.